Next speaker is, I think, requires no introduction, and Professor Nir Friedman as a professor of computer science and biology at the Hebrew University of Jerusalem. I think today we'll hear a very interesting talk. So if you can have the presentation up ready on chipstick of plasma cell-free nucleosome identifies cell of origin gene expression programs, and we look forward to your presentation. So, please.
Thank you for inviting me to give a talk, and I would like to tell you a story which is slightly different than RNA sequencing that we heard most of the day, but relates to single cell atlases. Hopefully you find it interesting. So as you probably know, cells die in our body all the time, and during a year of our lifetime, we probably lose about one cell per one body mass in terms of cells. And in pathology, many more cells are dying, and a lot of the cells that die inside our body essentially end up in the bloodstream. This is called cell-free DNA, and it's been used as a media for liquid biopsy in many situations, most notably to detect mutations in different types of cancers. The problem is that when we look at somatic cells, they are identical in terms of the DNA sequence. So looking at the DNA sequence does not differentiate a liver cell from a neuron cell or an epithelial cell. One thing that is slightly underappreciated in the community is that cell-free DNA is actually cell-free nucleosome. That is particles of histones wrapped around with DNA that come from the original chromatin template and circulate in the blood. Now, in the cell of origin, these histones carry what we call epigenetic marks that tell us a lot about the status of the DNA sequence that they are packing. And this suggests that we might, if we are able to read these, we might be able to gain a lot of information about what is those DNA sequences that we are getting. So to get this intuition slightly more concrete, if we're looking at epigenetic mark here using very abstract flags, if we have a red flag for suppressed and a green flag for induced promoter, then the same DNA sequence will be packed either in the brain cell as repressed, and in the liver cell, if this is an albumin promoter, as active. And if we can detect active albumin promoter, we know that it came from a liver cell. So if we can detect this nucleosome that comes from the albumin promoter that is also marked with active mark, we have evidence for where it came from. So the question is whether this is doable. And there are many barriers. The main one is that the plasma contains very little amount of nucleosomes, of DNA and nucleosomes, and it contains many other things, including many native antibodies in quantities that are much larger than we use in chromatin IP. We devised a method that overcomes this, and due to the lack of time, I'll skip the details, but try to convince you what we can do with it. So here is a standard genome browser for antibodies against 4 different marks. And the top 2 panels are 2 healthy donors plasma, and the bottom are from the Roadmap Epigenomics project. And you can see that the patterns are very similar. For brevity, we'll focus on just the top mark, histone 3 lysine 4 trimethylation, which is a mark of active promoters. And we can see that it appears at the promoter of GAPDH and other genes around it. And the question is, can we use that to learn something about what's going on inside the body? So quickly, just to convince ourselves what we learn from it, if we look at existing data, again, from Roadmap Epigenomics of K4 trimethylation, chromatin IP, and RNA, we see that for many genes, there is a very nice correlation between the level of that gene in different samples and the expression of that gene in the same sample. This is not true for all of them, but for a vast majority of the genes, this is indeed the case, suggesting that this is a good proxy for expression. So jumping into a much broader view, if we treat chromatin marks that promote K4 histone 3 lysine 4 trimethylation or K4 trimethylation as a proxy for expression, we can do a heat map similar to the way we do for gene expression. And here we use a diverse cohort of healthy subjects, cancer patient, various liver diseases, and acute myocardial infarction patients. And you can see that much of this picture contains a lot of very dynamic information, which I will not go into, but you can find in the paper I'm citing at the bottom. But I want to give you two quick views on how we can use this information. So we are thinking of what we see in the plasma as essentially telling us something about the activity of genes at the cell of origin. And so there are two scenarios we can consider, or a spectrum of scenarios that depend on how much that cell of origin of interest, say the tumor, is contributing to the plasma. On the one end of the scale is a situation that often happens in advanced cancer patients where a significant proportion of the cell-free DNA or cell-free chromatin that we extract come from the tissue of interest. And then we can treat it more or less the same way we would have treated direct chromatin IP from a tissue sample. For example, we can look at each gene and ask whether we see a level that is different than what we expect from the usual plasma patients, plasma in healthy people. So on the left, we see a healthy person compared to average of a different group of healthy people, and there are very little differences. Each dot is a promoter. And on the right, we see example from metastatic colorectal cancer patient, where there's thousands of genes whose promoters change by 100-fold or 30-fold. CDX1 is a classic marker of colorectal cancer, and EPCAM is a epithelial marker that is not present. It's very lowly present in healthy patients. And if we take this set of genes and do enrichment analysis, we get super significant enrichment to genes that are highly expressed in TCGA RNA measurements from colon cancers, colorectal adenocarcinoma. So clearly, this is real biology of the tumor. So what happens when we move from one sample to look at a lot of samples? Basically, while healthy subjects, and this is almost 100 samples, are all very highly correlated to each other, when we look at cancer subjects, we see very big differences in terms of, in terms of the correlation between samples. Nonetheless, there is a very clear signature of colorectal cancers. And again, I won't go into details, but we can use it to classify almost perfectly the samples, even when the cancer contribution is very low. But what are the sources of these variations? So one sort of variation between different samples of the same patient a long time are due to essentially changes in the amount of tumor contribution as the treatment succeeds or fails to tackle the disease. But when we look at interpatient differences, here are comparison of the same patient we saw before against two other patients that have more or less similar tumor loads in terms of cell-free DNA. And you can see that they are very, very different in terms of the many genes. And these genes reflect essentially differences in subtypes of cancer. And again, I'm skipping a lot of details, which essentially tell us information that is relevant to the clinician in terms of treating this cancer, because we can give molecular subtyping from this information. Skipping ahead, because I'm starting to run out of time, when we look at healthy subjects, when we look at low contribution, the situation is different. We cannot expect to look at each gene separately, but we might still see a lot of information about things that are expressed at very low level. For example, liver cells are expressed in 1 to 3% in healthy subjects. So can we see liver cells? So we devised a method where we use reference atlases to define signatures of places that are unique to a cell type or a tissue. And then we test that in our samples and compare it to background distribution. And we see the expected cell types in a healthy subject. These are many healthy subjects. And here is the amount of significant hits in different subjects. So all subjects, we see neutrophil and monocytes and megakaryocytes, which is a new discovery, which I won't go into. And we see very low level of liver and T cell, but these are significant levels. While heart and brain, for example, are totally not detected here in these samples. What happened when we switch from healthy subject to subject suffering acute myocardial infarction, where we know there is damage to the heart muscle? There we detect a significant level of heart signature, and this allows us essentially to say this is a controlled, it's a positive control that we can actually look at. Interestingly, if we actually try to identify the amount of heart contribution using DNA methylation CpG patterns, which again, I'm skipping a lot of details, you can see that the percentiles are very, very low, which raises the question, should we expect to see 0.25% contribution and detect it with significant level? We did analysis, turned out the math of essentially what levels of chromatin we should detect compared to essentially how many locations in the genome are informative. And you can see that for hearts, we more or less expect to see down to 0.1%. For more complex signatures such as cancer and so on, we can actually go much lower. So this is a very sensitive method. So the same way we detect changes in cell types, we can detect changes in transcription program or groups of genes that are target of transcription factors. And again, for the sake of time, I'm skipping a lot of stories here, and I'll just summarize that we have a new assay for cell-free chromatin. It's very robust and reproducible. It shows that those histone modifications are preserved through the cell death and through the circulation and the collection. And this has been, the last 3 years, we already did more than thousands of samples, and we're from multiple type of pathologies. And this is interesting both for the obvious clinical applications such as diagnosis, monitoring progress of disease, evaluating drug efficacy before the phenotype is actually viewable in imaging, but also a lot of basic science to understand what's going on inside our body at different situations. I'll end with the challenge where I think the connection to HCA is very strong. While in the body, everything is compartmentalized to cells, we are measuring something that resembles the fruit shake analogy in many of the HCA early presentation, where everything is mixed up together. And we need to essentially disentangle and talk about the status of these cells. And this is where good reference about the multitude of cell types, what are the expression in these cell types under different conditions, can help in terms of doing essentially guided analysis to get to this conclusion. And this is one of our efforts to try to integrate these two modalities that are not exactly the same, but they are connected to each other and hopefully can tell us about what's going on in the body. So I would like to thank again the organizers, my team, and many collaborators that were involved in this work. Thank you.
Thank you very much, Dr. Friedman. And it was fascinating discovery, and being able to understand the technology and apply it in clinical is beyond interesting of many researchers and clinicians.